Welcome to this tutorial about neurotransmitters. We continue to talk about our core concept about how neurons communicate through both electrical and chemical signals. And last time we spent our tutorial talking about the nature of the chemical signals, the neurotransmitters. Now we're going to talk about the receptors that they interact with. So we have some learning objectives today. I want you to be able to discuss the means by which ligand-gated ion channels affect the membrane potential of postsynaptic neurons. I want you to be able to compare and to contrast the structure and function of the ligand-gated ion channels and the metabotropic receptors, also known as the G-protein coupled receptors. We're going to focus on one very particular ligand-gated ion channel that's very important, especially in the context of learning about neuroplasticity. And I want you to discuss the properties of the NMDA receptor for glutamate and why it's so important for synaptic plasticity. And lastly, I want you to be able to once again recount for the factors that determine the effect of a neurotransmitter on a postsynaptic neuron. Okay, well, let's get started again by reviewing the basic structure and functional properties of a chemical synapse. And so those are recounted for you again in figure 5.3, a very important figure from your textbook. And what we see here is what happens when an action potential invades the presynaptic terminal membrane. So there's a wave of depolarization that causes voltage-gated calcium channels to open. Calcium rushes in to the presynaptic terminal, interacts with synaptotagmin. That causes the snare complex to pull together the membranes of the synaptic vesicle and the presynaptic terminal. That generates a fusion pore, and neurotransmitter then can diffuse out of the synaptic vesicle, where it can interact with neurotransmitter receptors on the postsynaptic surface. That's what we're going to talk about mainly in this tutorial. And so what we want to understand then are what are the consequences for activation of a receptor for a neurotransmitter on a postsynaptic cell. All right, so that's the challenge for the day. And I'll just remind you again that we have two classes of neurotransmitters, our small molecule neurotransmitters and then our peptide neurotransmitters. And these classes of neurotransmitters interact with two broad classes of receptors. So we will talk in some length about each class. We will begin with talking about our ionotropic receptors, which are receptors for neurotransmitters that are part of an ion channel complex. Hence we call these ligand-gated ion channels. And there are metabotropic receptors, which are receptors for neurotransmitters that are not directly part of ion channels, but rather these receptors trigger a set of metabolic activities that then might ultimately result in the opening or closing of separate ion channel molecules, as well as a variety of other postsynaptic activities. Our small molecule neurotransmitters typically interact with both ionotropic and metabotropic receptors, although that may not be universally true of all small molecule transmitters. Certainly our major important neurotransmitters like glutamate and GABA interact with receptors from both categories, whereas our peptide neurotransmitters interact with metabotropic receptors. Okay, so let's begin to consider what exactly these receptors are and consider their structure and functional properties. So let's begin by considering the ligand-gated ion channel. So as I said, the essential relationship here to understand is that the actual receptor sites themselves that interact with the neurotransmitters are part of an ion channel complex. Okay? So the receptor is part of the same molecular structure that forms the ion channel. And because of this, these are the receptor interactions that mediate the most rapid effects on postsynaptic currents. So as we go, I'll remind you that in order to understand the effect of any given neurotransmitter substance, whatever it may be, what we really need to know is something about the receptor to which that neurotransmitter binds. And we need to know something about what other receptors might also be present in the same postsynaptic cell in order to understand the physiology of that transmitter molecule. So we'll see examples of that as we go along here in this tutorial. All right, so let's consider the general molecular structure of ligand-gated ion channels, and we'll use the ligand-gated ion channel that we know best as an example. That would be the nicotinic acetylcholine receptor. So this is the receptor for the endogenous neurotransmitter acetylcholine that is also occupied and activated by nicotine. So this structure has a characteristic set of subunits that come together to comprise this ionotropic receptor. Typically, there are 5 subunits that come together to form the functional nicotinic acetylcholine receptor. Each subunit has an N-terminus domain to it that binds the actual neurotransmitter acetylcholine. So the N-terminus is out here somewhere in the extracellular space. Attached to that N-terminus is some number of membrane-spanning helical regions, and then there is some kind of cytoplasmic extension that often is a site where the receptor subunits can interact with metabolites and other kinds of molecules within the cytoplasm of the cell. So that's just one subunit. A functional channel requires the aggregation of multiple subunits, typically 4 or 5. Here in this example, we see 5 subunits that have aggregated, 2 alpha subunits, a beta subunit, a delta, and a gamma subunit. And they all differ slightly in their molecular structures. But together they aggregate to form a central pore that contains the filters that result in the selectivity of this ion channel. Now, as a class, ionotropic receptors are not as selective as our voltage-gated ion channels are. So as we'll see, multiple ions can pass through the pore of this channel. Now, what makes this a ligand-gated ion channel is the fact that channel opening is gated by the binding of the neurotransmitter to its receptor. So this ion channel is not particularly voltage sensitive. It doesn't have a voltage sensor motif among its membrane spanning domains. Rather, the gating mechanism is modified by the interaction of the transmitter with the receptor. That induces a conformational change that opens the pore of the channel, allowing for the passage of ions. Now, there are a variety of important examples of ionotropic receptors that we'll be talking about throughout the course. I've just been talking about the nicotinic acetylcholine receptor. There's a family of receptors for glutamate that are in the ionotropic category. There's a receptor named AMPA, AMPA, for the pharmacological agent that best activates that receptor. Likewise, there is a receptor called the NMDA receptor, N-methyl-D-aspartate receptor, and another one called the kainate receptor, all named for the exogenous ligands. Of course, the endogenous ligand for all 3 is glutamate. There's the receptor for the principal inhibitory neurotransmitter of the brain called GABA, gamma-aminobutyric acid. There are actually a variety of GABA receptors. Here are shown some of the subunits that come together to form what's called the GABA-A receptor channel complex, and so on. Receptors for glycine, for serotonin, and then for the purinergic transmitters such as adenosine. Now, what you'll notice from this chart here is that there are multiple subunits that can come together to form an ion channel, and combinations of subunits can form channels that have slightly different kinetic properties with respect to channel opening and closing, or perhaps different kinds of modulatory sites that are presented to the cytoplasmic surface, or perhaps different affinities for the endogenous ligands. So one can have different kinds of functional properties result from the aggregation of different kinds of subunits that come together to form the entire receptor channel complex. let's look a bit more closely at one important ionotropic receptor, and that is the receptor for the inhibitory neurotransmitter GABA. So what we're looking at here is the typical GABA A receptor channel, and it's comprised of 5 subunits that come together. In this case, an alpha, 2 beta subunits, a gamma, perhaps a delta or another alpha that have come together here. And like the nicotinic acetylcholine receptor, these 5 subunits aggregate to form a pore through which ions may pass. But what I want you to notice here is really the complexity of this ion channel. There's a variety of binding sites for other substances that can modulate the efficiency at